Welcome back to The Explainer. So, if you're hanging out with me today, you probably already know that plant biochemistry is just packed with hidden complexities. But today we're looking at something absolutely wild. It's essentially a high-stakes biological heist happening right inside the cellular walls of everyday broccoli. We're going to break down the precise mechanical and chemical pathways that create one of the most highly valued bioactive compounds in the whole pharmaceutical industry, sulforaphane. Okay, let's dive into this. Now, to understand how we actually extract sulforaphane in a lab setting, we've got to start with a pretty fundamental question. How does a simple, completely stationary plant like broccoli defend itself out in the wild against hungry predators? I mean, it doesn't have claws, it doesn't have teeth, right? So what exactly is its secret defense system? Well, the answer is a highly sophisticated booby trap, literally. See, the compound we're after here, sulforaphane, doesn't just sit there in the plant, waiting around for us to harvest it. It actually does not exist at all in an intact, healthy piece of broccoli. Nope. Instead, it has to be dynamically and honestly, violently triggered. Biologists actually have a great name for this. They affectionately call this brilliant piece of explosive cellular machinery the mustard oil bomb. It is easily one of the most fascinating biological mechanisms in the entire plant kingdom, for sure. And the craziest part? Its effectiveness relies entirely on one super simple principle: physical separation. So, to really grasp this physical separation, we need to zoom way in and look at the anatomy of a single broccoli cell. Section 1. Inside the plant vault, cellular anatomy. And this brilliantly illustrates exactly how the intact plant keeps itself safe. Deep inside the cell, there's this large water-filled organelle called the vacuole. Think of this as the storage vault. It safely locks away these sulfur-containing compounds called glucosinolates. As long as they stay inside this watery vault, they are totally inert, completely harmless to the plant. But floating right outside that vault, out in the cytoplasm, or sometimes even in entirely separate neighboring cells, is a very specific enzyme called myrosinase. This right here is the detonator. Because the vault keeps those valuable precursors strictly isolated from the detonator, the living plant doesn't, accidentally trigger its own chemical reaction and blow itself up. So if they're kept carefully separated, how does this booby trap actually go off in the real world? Section 2. Triggering the reaction, mechanical destruction. It turns out the trigger is purely mechanical. Physical tissue damage is the absolute key to setting this whole thing off. Whether it's a caterpillar taking a big bite out of a leaf, a farmer slicing a stem with a harvesting knife, or even just a massive tractor crushing the stalk out in the field, the structural integrity of the plant has to be destroyed. And when that physical destruction happens, total chaos at the cellular level. The cell walls break open, the vacuole ruptures, those previously locked away glucosinolates spill out and instantly mix with the myrosinase enzyme, the detonator. Because all this is happening in the presence of the plant's natural water, a rapid chemical reaction called hydrolysis kicks off. Instantly, those inert precursors are converted into isothiocyanates, like our prize, sulforaphane. It's incredibly bitter, really spicy, which immediately makes that caterpillar regret its life choices and back off. But it also just so happens to possess really powerful antioxidant and antimicrobial properties. This exact biological reality dictates literally everything about how we interact with broccoli in a lab. If you put on your agro-industrial engineer's hat for a second, you'll quickly realize you can't just gently soak intact broccoli stems in a solvent and expect a high yield. Gentle soaking leaves those vaults perfectly intact. To get the payload, we have to aggressively mimic that caterpillar. Section 3, the extraction protocol, mimicking destruction. Because we absolutely have to force these tiny cellular compartments to mix and generate these highly bioactive compounds, we need a very deliberate protocol. Let's walk through a standard 4-step extraction attempt. We've got slicing, drying, pulverizing, and finally an ethanol extraction. Let's see how this builds our chemical profile step by step. So step 1 is slicing the fresh stems down to about 5 millimeters. This is the initial spark. By slicing up that tissue, you are successfully breaking those plant cells. The vacuole opens, the myrosinase meets the glucoraphanin precursor right there in the plant's natural water, and boom, you've successfully created your very first batch of sulforaphane right then and there. Next up, the sliced tissue goes straight into a forced air oven at 45 degrees Celsius for 48 hours to dry. Now, what's really interesting about this slide is the trade-off. 45 degrees is actually a fantastic temperature to preserve the myrosinase enzyme. It doesn't denature and die until it hits around 60 to 70 degrees. You also successfully save the unreacted glucosinolates. However, sulforaphane itself is highly volatile. Leaving it baking in a forced air oven for 2 days means a significant portion of the sulforaphanate you literally just created in step 1, yeah, it likely just evaporated right up into the air. Then we move on to step 3, pulverizing the dried tissue into a super fine powder. At this stage, you are totally smashing all the remaining cell walls. The myrosinase detonator and the glucosinolate precursors are now perfectly mixed together in this dry powder. But remember, they need water to react. Because the powder is completely dehydrated from the oven, the reaction is entirely paused. The bomb components are sitting right next to each other, just waiting for a liquid trigger. Which brings us to the liquid trigger in the lab. But spoiler alert, this is where the plot twists. Section 4. The ethanol bottleneck, a hostile environment. To extract the compounds from that dry powder, the lab protocol calls for adding 60 milliliters of 80% aqueous ethanol. So the crucial point here is, the very moment that liquid touches the powder, the reaction completely stops. Done. While the 20% water in that mixture is technically enough to dissolve the compounds, an 80% alcohol concentration is severely hostile to biology. It's high enough to immediately precipitate and denature the myrosinase enzyme. The alcohol just instantly kills the enzyme's structure. And because the detonator is completely deactivated the second the liquid touches the powder, it cannot convert any of the remaining stored glucosinolates into sulforaphane. Section 5. The final chemical yield. Assessing the results. Okay, so after a final step of rotary evaporation and lyophilization, which is basically just freeze-drying the extracts safely at 40 to 45 degrees to lock in whatever compounds actually made it into the liquid, what do we actually have? Let's quickly summarize this crazy chemical journey. During slicing, sulforaphane was successfully created. Yay! During drying, unfortunately, much of it just evaporated right into the air. During pulverizing, the reaction components were perfectly mixed, but they were paused due to the lack of water. And finally, during the ethanol extraction, the whole reaction was permanently halted because the enzyme was destroyed. Now, if you're an extraction engineer, you might be kind of panicking right now, but don't. The protocol isn't ruined, it just dramatically shifted the chemical profile of our final extract. Because the 80% ethanol killed the enzyme, you actually did an incredible job extracting the intact precursor compound, glucoraphanin. And in the pharmaceutical world, this is highly valuable. In fact, many top-tier supplements sell pure glucoraphanin extracts. Why? Because they're relying on the human gut microbiome. Our own stomachs contain myrosinase-like bacteria that act as the perfect biological detonator to cleverly convert that precursor into sulforaphane later on, right inside our bodies. Which leaves us with one final, pretty provocative question. When you see broccoli extracts sitting on a supplement shelf, do the manufacturers actually have the commercial standards to test for both the precursor and the final product via an HPLC, a high-performance liquid chromatograph? Because honestly, proving the exact ratio of what was captured versus what was totally halted is the only way to know if you're buying a dud or a true mustard oil bomb. It's a huge testament to the incredible engineering hidden inside a simple vegetable, and it's exactly why we need to deeply understand the biology before we apply the chemistry. Thanks for joining me on this explainer, and I'll catch you in the next one.